Hi, I'm Mark Donald Dugay, graduate student of Grand Canyon University. So I'll be presenting on action potential of cardiac contractile cells. So let's start. So cardiac contractile cells are specialized cells which conduct impulses and are responsible for contractions that pump blood through the body. So the action potentials in cardiac contractile cells are normally initiated by the nodal pacemaker cells, which vary in ionic mechanisms and shape from the sinoatrial or the SA node potential. So the figure shows the action potential in cardiac contractile cells, which remains essentially at rest about 90 millivolts until it is excited by an electrical activity propagated from the SA node, which is basically the natural pacemaker of the heart. So once the cardiac contractile cells are excited, the action potential rapidly rises due to the sodium entry and opening of fast sodium channels at threshold. So this phase, depolarization occurs because of the opening of the fast sodium channels where potassium flux also decreases. After the potential reaches its peak, a brief repolarization occurs as a result of limited potassium flux and opening of transient potassium channels coupled with inactivation of the sodium channels. So the plateau phase occurs, which is unique to the cardiac contractile cells. So this prolonged plateau is maintained by two voltage-dependent permeability changes. We have the activation of slow L-type or long-lasting type calcium channels coupled with a decrease in potassium permeability in the cardiac contractile cell membrane. So the movement of calcium ions out of the cell in this phase maintains depolarization. So the rapid falling phase of the action potential results from inactivation of calcium channels and delayed activation of ordinary voltage gated potassium channels. The decrease in calcium permeability declines the low inward movement of calcium. So this occurrence simultaneously increases potassium permeability, which promotes rapid outward flow of potassium. So then the potassium leaves the cell where it returns to resting potential. So this state is maintained by the reopening of leaky potassium channels and losing and closing of the ordinary voltage gated potassium channels. So that is the action potential of cardiac contractile cells, whereby the distinct feature of this um, action potential, okay, the has a large stable resting membrane potential and displays a prolonged action potential with the plateau phase. So development of therapies to improve heart function, relieve symptoms, reduce hospitalization, and improve survival is a high priority in cardiovascular medicine. So in this scholarly source titled Cardiac Contractility Modulation by Non-Excitatory Currents, Studies in Isolated Cardiac Muscle, is related to the content of my presentation or the assignment that I have presented. So the effect of cardiac contractility modulation by non-excitatory currents in isolated cardiac muscles were evaluated. So the control group, okay, was the rabbit right ventricle papillary muscle, and the experimental group was the human failing trabecular muscle. Identical methods were used to isolate, prepare, and study the muscles obtained from these two organisms. So figure 1 shows the methods used in papillary muscles whereby the muscle strips, so specifically the papillary muscles and the human filling trabecular muscle obtained were placed in an organ bath or the muscle bath and were subjected with carbon electrode. Okay, then the CCM signals were delivered separately. So the CCM electrode and two reference electrodes, G1 and G2, okay, these signals caused muscle contractions were force generated and measured using the force transducer, and transmembrane potentials were recorded using micropipette. So again, so this is the method used, okay, to test or to evaluate the effect of CCM modulation by non-excitatory currents isolated from these two organisms. So figure 2 presents the effect of cathodic ANC and the anodic BND CCM stimuli on isometrically contracting rabbit papillary muscle. So figure 2 and A and C show the effect of cathodic CCM on rabbit papillary muscle generated less force, which caused shorter muscle contraction. While figure 2B and D, the effect of anodic CCM on rabbit papillary muscle generated more force, which caused longer muscle contractions. So this means that the negative amplitude or the cathodic CCM induce early repolarization and weaken the heart's contractions, which slow down the heart rate, or there is what we call negative inotropic effects. Okay, while the positive amplitude or the anodic CCM enhance contractility due to prolonged depolarization. Okay, figure 3 shows the effect of CCM signal parameters, which include specifically the delay, duration, amplitude, an action potential duration or APD, and developed force in rabbit papillary muscle. Okay, and the sample here is 6. As shown in figure 3, so the effect of CCM signal parameters, duration, amplitude on APD varies with its effect on the developed force or the enhancement in rabbit papillary muscles. So the effects of CCM signal parameters, okay, the delay, duration, and amplitude, and APD are comparable with the effects of CCM signal parameters on developed or enhancement, whereby on ADP, the effect of CCM signal delay with duration of 30 milliseconds. So the amplitude of 3.5 milliampere, or duration with delay 30 milliseconds and has an amplitude of 3.5 MA. While on developed force, so the effect of CCM's signal delay with duration of 20 ms, amplitude of 4.5 MA, or duration with delay 40 ms and amplitude of 4.5 MA. So on the other hand, so the effect of CCM signal amplitude and APT and developed force, okay, for B and D is the same with delay 30 ms and duration of 30 ms. So figure 4 is the involvement of sarcoplasmic reticulum in the CCM effect. So figure 4A shows the effect on papillary muscle exposed to ryanodine before and after. So the gray tracing represents before, okay, treatment, and the red tracing represents after treatment. So ryanodine ion channels that are responsible for the release of calcium from the sarcoplasmic reticulum. So it is obvious that there is a decreased contractile force, okay, with or without CCM signal. Okay, okay, figure 4b showed the effect on papillary muscle with and without CCM signal, where strong contractions were generated without CCM signal compared with the CCM signal. All right, so figure 5 shows the effect of anodic and cathodic CCM signals on human trabeculae. So basically, human trabeculae are muscular columns which project from the inner surface of the right and left ventricle of the heart. So the effects are similar to the observed in rabbit papillary muscle, whereby the anodic CCM, which is the positive polarity signals, increased contractility, where the cathodic CCM, the negative polarity signals, depressed contractility. So based from the results of this study, so the effect of cardiac contractility modulation or the CCM by non-excitatory currents provided the potential as a therapeutic strategy which enhanced contractility, particularly in patients with heart failure. So that ends the presentation and here are my references. Okay, mahalo.